Hi, my name is Cambria Revson, and my project is titled Changes in DNA Methylation of Salt-Treated Brassica rapa. A large problem in agriculture today is that a high percentage of land we could be using to farm crops is unable to be used because there's too much salt in the soil that prevents crop growth. And a large amount of money, $27.3 billion, is wasted every year in unsuccessful attempts to solve this problem. So this obviously leads to a decrease in the production of food and materials that we need in our daily lives. And there are several solutions to this problem, but the one I will be arguing today is the modification of plants' epigenetic responses. What is epigenetics? Epigenetics is the response of organisms to environmental stressors. So this can be temperature, food, etc. And the most prevalent form of epigenetic responses is DNA methylation, which just includes methyl groups, which are groups of 1 carbon and 3 hydrogens that attach to the cytosines of DNA. And the amount of these methyl groups determine a gene's expression. So in the promoter region of a gene, if there's more methyl groups on this specific gene, the gene will be higher expressed, and if there are fewer groups, the gene will be lower expressed. My thesis for my project was sodium chloride applied to Brassica rapa plant groups will increase the methylation on the gene BRERF4, promoting its expression and thus resulting in heightened stress response in the plants to salinity. So I looked at this one specific gene in Brassica rapa, and this gene codes for salt resistance and stress response in Brassica rapa. So I wanted to look at how it would change in terms of different levels of salt and how this would affect the DNA methylation. So I had a couple of objectives for my project. I wanted to see the effect of these different sodium chloride concentrations on my plants in terms of phenotype, DNA methylation of the gene, and expression of the gene. So I started off my project by planting Brassica rapa seeds to grow 4 different plants, and then I added my salt treatments in increasing concentrations from no salt in the water to the most salt. And I took daily measurements of my plants for a number of different factors: height, width, leaf count, and then I analyzed these differences in measurements using t-tests. And then for the second part of my process, which was more the DNA analysis part, I first designed a primer for my BRERF4 gene, and then I extracted DNA samples from my 4 different plant groups, and then next I applied HPA2 to the DNA samples, and this is a methylation-sensitive restriction enzyme, which means that it was able to cut unmethylated DNA but not methylated DNA. And then after this had ended, I performed PCR, polymerase chain reaction, on my 4 different samples, and I analyzed my results to provide conclusions about the methylation. My results were in 2 different parts. First, for the t-tests, and this was just looking at the results of the measurements that I had taken over time of the plant's growth. I found, as one might expect, that there was a significant difference between the plants that had received the most salt and the control group, which had not received any salt. And this just confirmed what I had already expected, which was that salt inhibits the growth of Brassica rapa. And then the second part of my results were in the PCR, and I looked at the rate of amplification of my 4 different DNA samples from my control group, my slight salt concentration group, my moderate salt concentration group, and my strongest salt concentration group, and those are in order in the graph shown. So what I found was that there was a higher rate of DNA amplification of my slight and moderate salt groups, those are the middle ones, as compared to my control group. And I knew that DNA, when it's amplified quicker and easier in PCR, this is because the strands are physically longer. So in my slight and moderate salt concentration groups, this suggested that the DNA had been cut less by the methylation-sensitive restriction enzyme, which meant that it had more methylation. And vice versa in my control group, since it was cut more, this meant that it had no methylation. And this didn't continue on in my strongest salt concentration group for unknown reasons. But basically what this showed was that the methylation of my slight and salt concentration groups was increased, which showed that the gene was higher expressed because it had more of a need to respond to the salt.